How's it going, everyone? So today in this lecture, we're going to talk about both lipid metabolism and protein metabolism. And the reason that we're going to put both of these in one lecture is because they both sort of end up in the same place that carbohydrate metabolism does. They just have a few steps before they get to something that you're already going to be familiar with. So let's get right into it. The first thing we're going to talk about is what triglycerides are. So they're fats, and they're ingested through your food, or they could be synthesized from your adipocytes, which are your fat cells, from carbohydrate precursors. So you can sort of make these triglycerides from carbohydrates. Lipid metabolisms entail the oxidation, which remember is loss of electrons, of fatty acids to either generate energy, like what we're going to talk about, or to synthesize new lipids from smaller constituent molecules. Lipid metabolism is associated with carbohydrate metabolism, as products of glucose, like acetyl coenzyme A, can be converted into lipids. Lipid metabolism begins in the small intestine, right? Because we don't have any enzymes that are going to be digesting these before we get into our small intestine. We get these ingested triglycerides that are going to be broken down into smaller chains of fatty acids and then subsequently into monoglyceride molecules by pancreatic secreted lipase. And that happens after they've been emulsified by bile salts. Because remember, these fats, basically, they will stick together in aqueous environments because they're very hydrophobic. And so bile needs to emulsify them so they don't stick together and that they can move through the aqueous environment. The pancreas is stimulated to secrete lipase by a hormone called cholecystokinin. Cholecystokinin. Together, the pancreatic lipases and bile salts will break down triglycerides into free fatty acids. And those fatty acids then can be transported across your intestinal membrane. And then once they're across the membrane, they recombine again into triglyceride molecules. Within the cells, once they've gotten into cells, those triglycerides are packaged along with cholesterol molecules in phospholipid vesicles called chylomicrons, and that's what you see on the left here. This is a chylomicron. And the chylomicrons basically enable fats and cholesterol to move within the aqueous environment of your lymphatic and circulatory systems, kind of exactly like what bile did. It's just so that they don't get stuck and coagulate in a certain area. Once you get into the circulatory system, they can either go to the liver or be stored in fat cells, adipocytes, that comprise your adipose or your fat tissue found throughout the body. Let's talk about lipolysis or lipolysis. So what does it sound like? It sounds like glycolysis. It's basically this cutting up of lipids. To obtain energy from fat, so if we're going to use the fat not as a storage but as an energy source, you have to first break down triglycerides by hydrolysis into their two principal components, which are the 3 fatty acids and a glycerol. That process takes place in the cytosol just like glycolysis does. The resulting fatty acids are then oxidized by beta oxidation into, what do we know, acetyl coenzyme A. What do we know about acetyl coenzyme A? It's the very first thing that joins up with the 4-carbon molecule oxaloacetate and makes citric acid into the Krebs cycle. The glycerol that's released from the triglycerides actually enters glycolysis as DHAP, which, if you remember, is just a 3-carbon molecule that has to be changed a little bit before it could become G3P. So, because one triglyceride molecule yields 3 fatty acid molecules that could have 16 or more carbons in each one, if you think about what a glucose is, it's 6 carbons, so more carbons, more energy. Fat molecules actually yield more energy than carbohydrates and are an extremely important source of energy in the human body. We're not really going to just be getting along very well if we're just intaking glucose. And in fact, triglycerides yield more than twice the energy per unit mass when compared to carbs and proteins. So therefore, when glucose levels are low, triglycerides can be converted into acetyl coenzyme A and used to generate ATP through what we just talked about, the arrest of aerobic respiration. Okay, let's talk about ketogenesis. If you have excessive acetyl coenzyme A, and that would be created from the oxidation of fatty acids, and you're just bringing in a lot of material into your diet, um, the Krebs cycle could get overloaded and not be able to handle it. And so then the acetyl coenzyme A gets diverted to create what are known as ketone bodies or ketones. And ketones can serve as a fuel source if glucose levels ever get too low in the body. Ketones serve as fuel in times of prolonged starvation, or when patients might suffer from uncontrolled diabetes, or if you're familiar with diets like the Atkins diet, is just the common name for going into ketosis. It's basically eating a very small amount of carbohydrates so that your body then goes into ketosis and uses ketones as a fuel source. In both of those cases, or well, in the three of the cases that we've talked about, fat stores get liberated to generate energy through the Krebs cycle, and it'll generate ketone bodies when you get too much acetyl coenzyme A. So basically what happens is you starve your body of easy to obtain glucose, and then it has to go into your fat stores to get acetyl coenzyme A and make basically ATP at the end of it. And so a lot of people like going on diets like this because basically you can pretty much eat all the fat that you want. You just don't eat carbohydrates and processed starches like that. And you eat a lot of protein, and it basically starts to melt your fat off. Obviously, that is easier said than done, basing on how we eat in this country, where everything is so driven around starch, but it is effective. Up to this point, I've been telling you that neurons really only are able to use glucose as their energy source. But the brain actually can use ketones as an alternative energy source. The brain will keep functioning when glucose is limited, and when ketones are produced faster than glucose, they can be used, and then they can be broken down into CO2 and acetone when you have too much. And the acetone gets removed every time you breathe out. And so one symptom of being in ketogenesis, or ketosis basically, is that the patient's breath, or just someone who's in ketosis, their breath will smell sweet. And actually that's one of the ways that somebody first described diabetes, is because they could smell their breath. It's actually a really big sign of having diabetes. And then the carbon dioxide produced could acidify your blood, leading to diabetic ketoacidosis, which is a dangerous condition as well. Okay, let's talk about lipogenesis. When glucose levels are plentiful, the excess acetyl coenzyme A that might be generated by glycolysis is then converted into fatty acids, triglycerides, cholesterol, steroids, bile salts, and that's a process called lipogenesis. It basically creates fat from the acetyl coenzyme A that you can't use because your Krebs cycle's got plenty of it. And it takes place in the cytoplasm of adipocytes, which are fat cells, and hepatocytes, which are liver cells. When you eat more glucose or carbohydrates than your body needs, everyone's pretty much aware of this, but your system uses acetyl coenzyme A and turns it into excess fat. So sad. And then let's get into protein metabolism now. So proteins, we know they make up amino acids. They're made up of amino acids, and they make up a ton of structures in the body. Enzymes, cell signaling receptors, signaling molecules, structural members, intracellular trafficking components, extracellular matrix scaffolds, ion pumps, ion channels, hemoglobin. Protein is also used for growth and repair of cells and cell structures. And amidst all of that, you can actually, of course, use protein as an energy source. Proteins don't get stored for later use. So you either use them up, and if you've got any excess, they have to be converted into glucose or triglycerides and used as an energy supply, or obviously put into some sort of energy reserve. And although the body can synthesize proteins from amino acids, you have to eat a lot of these amino acids because we're unable to synthesize all 20 amino acids that are used to build proteins. The digestion of proteins obviously begins in the stomach because you can get protein-rich foods that enter the stomach and they encounter a very acidic environment there. You get an enzyme pepsin and hydrochloric acid. Hydrochloric acid is an acid. It has a pH of 1.5 to 3.5. And when you have an environment that is extremely acidic, or is just something that is not the ideal environment for a specific enzyme, that enzyme loses its shape, or what is said here, becomes denatured. And then the enzyme pepsin cuts proteins into small polypeptides, or basically just amino acids. And then when you get the chyme that enters the small intestine, the pancreas releases sodium bicarbonate, which neutralizes the HCl so you don't burn any of the rest of your body. And the small intestine also releases digestive hormones, including secreting CCK, which stimulates digestive processes to break down the proteins further, and secretin that stimulates the pancreas to release more sodium bicarbonate. And then the pancreas will release most of the digestive enzymes, which also aid in protein digestion. All of that together, those enzymes break down complex proteins into the small individual amino acids, which are then transported across the intestinal mucosa to be used to create new proteins, or to be converted into fats, or of course acetyl coenzyme A, which is used in the Krebs cycle. The last thing is the urea cycle. And when you have freely available amino acids, you want to use them to make proteins. But if you have too many and you can't use all of them, there's really no capacity or mechanism to store them. And so they have to be converted into glucose or ketones or be decomposed. When you decompose amino acids, you get hydrocarbons and nitrogenous waste. And basically, if you have too much nitrogenous waste, that becomes extremely toxic. The urea cycle is a process to process nitrogen and facilitate its excretion from the body. The urea cycle, it's a set of biochemical reactions. It produces urea from ammonium ions in order to prevent a toxic level of ammonium in the body. So ammonium is toxic, urea is not. And once you break down that ammonium, which occurs primarily in the liver and some in the kidneys, and of course, if you know much about that, that's where you're making urine. And prior to that, ammonium ions are produced from the breakdown of amino acids. In these reactions, an amine group or ammonium ion from the amino acids exchange with a keto group on another molecule. And basically, that's called a transamination event, and it creates a molecule that's necessary for the Krebs cycle, and then an ammonium ion that enters the urea cycle to be eliminated. And in the urea cycle, ammonium is combined with CO2, which makes urea, which is totally non-toxic, and water, which is of course non-toxic. And then you eliminate urea and water, of course, through the kidneys in your urine. And so that covers those two sections of that chapter. Let me know if you have any questions.